Hello everyone, welcome back. I hope you had a good lunch. Let's get into this. The talk of today, let me try and, okay. Full screen, can you see my full screen? Okay, let's try and share. Okay, share. This is something. Yep, okay, now full screen. Now full screen. Sorry guys, we're having this problem again. Okay, share. You see my PowerPoint here? Okay. Now I am. Let's see. Let's keep. So, sorry, give me a second.
Sorry guys, we have slight difficulties with screen sharing. We are getting there. Give us a second. Until then, obviously, I can entertain you a little bit with some things. Obviously, we have a website, and the website has, as before, all the presentations. So, and you find all these kind of tutorials which we run at the end of the workshop still there, and they will be there for eternity. Also with the files, some of them are bigger. We have to think about where we put them, but basically, all the tutorials can be run at a later stage. What else is there? Oh yeah, I have one thing that might be important. When you log into the Dart Cloud now, yeah, under your content, you should see now only this session, hopefully session 5.6, and you click on that, and then you should be able to make a copy from that. So basically what I will distribute, because we updated so regularly, we are not finished yet. Basically every session or maybe 2 sessions will have their own repository. So please check the dartRverse, and if you can't log in because you're losing a link, click here on the dartRverse workspace, and then, because it's me, I see a lot of them, but you probably will only see one of them and your old ones. So click on the one that says this is the one to copy from you. At the moment is session 5.6. And I think, are they ready? No. You wanna do it here, Diana? On another computer? Yeah, sorry guys, still a little bit of trouble here. We're frantically working on that. Maybe Should I put a poll out there to keep them entertained? I'm not sharing the screen. I know. At the moment, I'm also not there because there's some glitch that Diana can't share the screen for whatever reason. So we may have to resort to another computer. So give us another minute or two to sort that out. Because we have some time, we can obviously use it for some questions or some other things. So please put something in the chat and talk to us or raise your hand. You can do that, I think. Put you on the stage. Okay. Yeah.
Cannot leave. Wait, sorry. Okay.
Okay.
Sorry.
And then you can go back here.
Okay. Okay, I do this. Okay, and then get presenter view.
Yeah.
Okay. Presenter view, and then I do alt tab. It's this one. Oh wait, it worked. Okay. Then I do.
Back to
What can you see? Okay, go back to RMS. Share.
Yeah.
No. Which one?
I can check if Renee is there. I try to find her.
And then how do I go back? With this, like this.
And I can send her a message.
I go back. It's not there. Where is it? It's this one. Yeah, yeah, it worked. It works. It works. Yeah, yeah. Can someone unmute me?
Okay, I can't show you how excited Diana is at the moment because it works.
It doesn't
But
change.
I guess we're almost there, so I
It doesn't change?
Yeah, it's still not changing.
hand over to her in a sec. Is that okay?
No.
It doesn't. Yeah, it changed. Yes,
Okay.
yes, it works.
Okay. Okay, here we go.
Unmute me and turn on my camera.
No,
Yeah, we want to see this excited face.
Yeah.
Yeah.
Can someone turn on my camera, please?
No, just you can do it.
Only I can do. Okay, how can I do that? Okay.
Yeah.
Okay, almost there. Almost there. Okay.
There we go. Okay, there she is.
Wait. Getting there. Wait.
Still there.
Okay, does it work? Wait. Does it work?
Yes.
Yes. Yay. Oof. Yay.
Okay.
Welcome back.
So now, after all this beautiful intro, I couldn't have done it better. I hand over to Diana.
Okay, so before we start, I want to thank Ching Ching. She was a Jedi here and helped me troubleshoot this. Please, some claps for Ching Ching.
Thank you.
Ching Ching.
Hi.
Yay, awesome, perfect. Okay, after this hiccup, let's go into what we can do with small populations so that we don't manage them to extinction. I need to tell you that this is a whole field. We will just be able to scratch the surface in this session. As usual, it will be divided in two parts. First part, theory, that's given by myself. And this will not be exhaustive, but I want to leave you with some key concepts and some rule of thumb. And in the second part, Peter will guide you through a brief tutorial. Let's get into it then. In a galaxy far, far away, there was a conservation program that wanted to protect the dusky seaside sparrow. This is an American bird whose population plummeted until only a handful of individuals were left in the wild. The conservation program did huge conservation efforts. They had a really large budget and very fancy infrastructure. Despite all of this, the dusky-seaside sparrow was declared officially extinct just a decade later. This is our worst nightmare, right? They had everything they needed to save it, and they didn't. So what went wrong? Well, human activities have transformed natural habitats over the last few hundred years, and many species on the planet now have these fragmented distributions in which natural gene flow that was occurring in them has been interrupted. And that has left us with small, isolated populations. And there's specific ways in which evolutionary forces behave in these populations. So let's delve into the genetics of small populations. There's two important things that happen in small isolated populations. One of them is inbreeding. During inbreeding, which is when relatives mate with each other, the homozygosity on the genome increases. And that has the effect of lowering the fitness, partly because harmful alleles that are usually recessive get into homozygous state, and then they get to be expressed. And this effect of having higher heterozygosity and low fitness is called inbreeding depression. Inbreeding depression is a bit hard to measure. It is challenging to have a population and a sample large enough, but we have managed to do this for the helmeted honey eater. And you can see here in this plot in the x-axis homozygosity, and in the y-axis, the total fledglings that each bird produces over their lifetime. And this is really remarkable. These are long-lived birds. They can live up to 22 years. So here we have how many fledglings they produce over their whole life. So this negative relationship in which as birds get more inbred and more homozygous, they produce less fledglings. Inbreeding depression is typically more severe in stressful compared to benign conditions. So it's usually more, it's larger, the effect is larger in the wild compared to when you have captive populations and you have them all pampered and protected. And inbreeding depression is also more severe for total fitness measures than for individual components. For example, if you have, in this case, total fitness measure in which you have literally all the fledglings that were produced versus, let's say you have only how many eggs were produced in one breeding season, the effect will, of course, look larger and it will be larger in the total fitness one. For inbreeding depression, effective population size is crucial. And you will have a session in the next few days about how to calculate effective population size. But for example, let's compare this population, the big one in which we have 100 effective individuals, against 5 populations of only 20 individuals each. So in total, there's the same amount of individuals. These marmots, they have different inbreeding rates just because of the size. So in the large one, we have inbreeding rate of 0.5%, while in the inbreeding one, in the other one, we have 2.5%. And this might not sound like much, but after only 10 generations, the inbreeding in the large one is 4.9%, compared to 40% inbreeding in the smallest populations. And that is in part why effective population size is so crucial. The second important thing that happens is that small isolated populations lose genetic diversity really fast and very easily. And with this, they start to lose their evolutionary potential. So they have a reduced capacity to adapt. You can look at genetic variation kind of as a toolbox. The more, if you have very few tools in your toolbox as a population, the less likely it will be that one of them will give you the fitness advantage necessary to deal with environmental changes. So in this case, they are less likely to adapt. And this happens when a population goes through bottleneck. You have a lot of genetic variation, then something happens to the population in which the population size gets reduced severely, you lose alleles, you lose variation. And this effect of randomly losing genetic variation is called genetic drift. And typically, the thing that goes away first are the rare alleles, simply because they are less likely to be contained in the new small generation. So that's genetic drift. So in small populations, natural selection is a lot weaker than in large populations. Basically, natural selection kind of gets overwhelmed by genetic drift. So you can imagine that you have a very small population, and maybe due to the selective pressures, you kind of start to see some survival of the ones that have good traits, but something happens to that small population that kind of randomly just select some individuals, not based on the trait, but just at random. That's the thing with small populations. They are very likely to just the stochastic effects affecting them. So due to genetic drifts, these populations just start to diverge randomly. They start to accumulate different genetic variation over time. But this is not necessarily adaptive variation. It was just the effect of a stochastic. So remember this, because they will start to look different. Small populations will start to look different just by chance. How fast a population loses genetic diversity and starts to diverge from the others will be determined by its effective population size. So you can see here that FST is a measure of differentiation. You can see that populations with a size of 20 very quickly get more differentiated compared to a population that it's at least 500 individuals. So if we had to summarize the genetics of small populations, small isolated population fragments, is that they simultaneously become genetically differentiated, they lose genetic diversity, and they become more inbred. And well, in this case, size matters and connectivity matters. The smaller and more isolated a population is, the more severe these problems will be, and the higher risk they will be into, the faster all of these things will go. So in this sense, because small isolated populations have high inbreeding and low genetic diversity, these two factors greatly increase their extinction risk. So since a couple-ish decades ago, scientists have realized that saving these species and these populations from extinction requires, as a complement to all the traditional conservation measures like revegetation and captive breeding and all of those things, predator management, etc., it also requires the genetic management of threatened populations. We can intervene to restore the genetic mechanisms that would have normally kept the population healthy, but they have been disrupted in the deteriorated habitat. Namely, a very important one is gene flow, through the implementation of genetic rescue. Now, poll time. Can we please have the first poll, Bernd or Luis? What is genetic rescue? Poll, please. Or I don't know if Bernd, the poll, please. We are working to get the poll. What is genetic rescue? Correct.
Yeah.
All right. All right, then some votes. Yeah. 50 votes. Come on, come on. Place your bets. All right. I think we can start working with this. So there's different ways to define genetic rescue. One way in which we can do that is from the wildlife manager's perspective, is to say we are assisting gene flow from one population to another. But in the genetic population sense, genetic rescue is also defined as the benefits of that gene flow. So basically the resultant population growth that you get after bringing in gene flow. So both the second and the third options of the poll were correct. But in this case, I will be referring to genetic rescue as the movement, the assisting of gene flow into the population, just to make it easy. Right, so assisting gene flow into a genetically isolated population will be with the purpose of alleviating all these problems, inbreeding depression, and to restore genetic diversity. Genetic rescue has actually been implemented in a variety of plant, vertebrate, and invertebrate taxa. And although it has been found consistently successful and widely so, it remains still to be it is still underutilized, and mainly because there are some fears of having some unintended consequences from that gene flow. And that includes outbreeding depression. So very quickly, outbreeding depression is when you get reduced fitness in the offspring of the crosses between populations that are too different. So basically, if you mix them, will the offspring have lower fitness? We know that outbreeding depression is rare, and we know when it is likely to happen. It is likely to happen when the populations have diverged a long time ago, when there have accumulated important chromosomal differences, and when they have been adapted to different environments. So when you have these things, the crossings, the offspring will be likely to survive or to reproduce adequately. But because we know all of these things, outbreeding depression is predictable, and therefore it is avoidable. And in reality, it is actually pretty rare. So it is because of this that managers must systematically weigh the potential risks and benefits of doing genetic rescue, but also the risks and benefits of not doing genetic rescue, because not doing genetic rescue is also a choice. And if we fail to do this, many conservation initiatives can fail, just like the dusky-seaside sparrow one did. They were too afraid of mixing it for too long, and when they finally did, it was too late. The last female died, and they just saw the last males 4 years until they died. And that was it for the dusky-seaside sparrow. Fortunately, there are some amazing guidelines that are specifically targeted to conservation scientists and to wildlife managers. There are these 2 books that were designed specifically for that. And for this theory section, I am drawing very heavily from these books. Please always refer back to them. They are written by some of the giants of conservation genomics. Now, time for the second poll, Bernd or Luis. Second poll is, are you familiar with the work of these master Jedis? So Frangam, Rawls, Ballou, Lacey, or Polsonex? They are my personal heroes. All right, 64 votes. Cool, okay. A bit less than I expected, but all good. So there are guidelines, and some of them look like this. They are decision trees. This is a simplified version, so you can look at the books. But they are decision trees that have been built by these people based on they are experts in the field, and they have tons of decades that they have compiled together, tons of evidence to guide you through this process. So let's go. The first part, genetic assessment. How do you go about making management decisions? And we can have 2 parts for that. First is making a diagnosis, and second is to gene flow or not to gene flow. That is the question. So for the first part, for the diagnosis, in order to make the first diagnosis, is you would like to identify if there are genetically fragmented populations. That means that there is reduced gene flow between them. And this is kind of a gradient, right? So you can have on the left part, on A, a population that is very large and that is randomly mating. And then as you start going into the right, you get more isolation, and you get more isolation by distance. So you can start having a large population that is, however, isolated by distance, so the geography. Then you can have them split. Maybe there's a river, there are mountains, things that really isolate them. Then when your populations start to vary according to an ecological gradient, you can get this correlation that is called a cline. You can have fragmented clines, they are physically separate. And lastly, you can have fully isolated populations in which there is no gene flow according to them, and they have been genetically differentiated. So in summary, you are kind of trying to find if there are genetic clusters. As we have already seen in the previous sections, you can run a structure analysis, and we already saw how that goes about. You can also look into PCOAs and some differentiation measures like FST. But I need to also make a point, and that was mentioned in the previous section. For example, here we have an example. This cutie is the Leadbeater's possum, and it is the cutest mammal on Earth. I don't care if you disagree, fight me. We collected samples a few years ago, so you can see in the map the location of the samples that we selected. So when we draw a PCA, we saw these kind of discrete groups, and we were thinking, oh no, maybe they are getting really genetically differentiated. What are we gonna do? But later on, someone brought about that maybe it was due to our sampling that we are seeing those discrete units. So we went in, we got more samples, kind of in between the locations. And when we run a second PCA, then we find that it's more of an isolation by distance thing, that there seems to be some exchange in some populations. So the gray one is a specific case, that's Yellingbo, that's highly endangered. But for the rest, there seems to be more gene flow going into them. So keep in mind that your results will be only as good as your sampling design. And this is something that Renee talked about. This is something that Bill and the rest talked about. Sampling is key. Back in the day, when people saw that there was population structure and that there was some limited gene flow, they would decide to manage them separately because they look different, right? However, when these things happen, they are likely to be under genetic erosion, and that is the recipe for extinction. So you remember what we said, the small populations get differentiated at random, and not the genetic variation is not necessarily adaptive. So they get things, genetic variation fixed just by chance. So something typical of these strongly differentiated population fragments is low genetic diversity, high inbreeding and inbreeding depression. And we can see generally that the more differentiated a population is, so that's FST, the more inbred it is, the heterozygosity is lower. So if you find genetic differentiation in your data set, assess for genetic erosion. Always, always, always. I cannot emphasize this enough. Please don't just go and keep them separate because they look different. You have to assess for genetic erosion. Okay, you know better than to do that now. Okay, going back to our guidelines, let's say that we found differentiation in our populations. Now we follow the decision tree. The first step is let's look into genetic erosion. And for this, we assess for evidence for at least one of these ones, one or more. With that is enough. First, did you find in some of your small populations that they have lower fitness than the other larger ones that are non-inbred? Therefore, you are finding inbreeding depression. You can measure this with total fitness measures, but basically any fitness measure will do. Or did you find lower genetic diversity in the small populations than in the larger, more connected ones? You can measure this with heterozygosity, with allelic richness. Did you find that they have high inbreeding, higher than 10%? There's some tools that you can use. You can use kinship. You can use individual inbreeding coefficients. Or Have you found that they are losing genetic diversity, that they have lost 10% of their genetic diversity? Or do they have a small effective population size? Or have they had no gene flow for multiple generations? I need to emphasize that one of these is enough. Because if one of these is true, most likely the rest are also true, and you're in the face of some genetic erosion. Big note here. So a few years ago, it was kind of necessary to prove inbreeding depression, but that is really, really not necessary to do. First, we are working under urgency as conservation biology is a crisis discipline where inaction often leads to decline and extinction of threatened populations. And this is very hard to do, proving inbreeding depression. It takes a lot of time. It's difficult. And in the meantime, the clock is ticking. Second, we know that inbreeding depression is ubiquitous. It's everywhere. It happens to populations that are naturally outbreeding if they are in these conditions. And third, most experiments have too low of power to detect inbreeding and inbreeding depression. Conversely, there has been very large meta-analysis in which they have taken evidence from many, many years, from many, many studies, and they have found that inbreeding depression happens all the time. So, yes, time is of the essence. One of these is true, you are likely looking into genetic erosion. Let's go back to our guideline. The second step is there a potential donor population? And ideally, you would want the donor population to be of the same species, to not be a recent derivative. That means that if you had the big population, the donor population should not be like a subset of that one. And ideally, the donor population should be larger and more genetically diverse than to target one that is small and inbred. So try to find the better ones that you have at your disposal. Guidelines. Third, we need to weigh the risks of doing genetic rescue. So that's the risk of outbreeding depression. Yes, so outbreeding depression, lower fitness of the crosses. This is rare, as I mentioned before. This happens when there's fixed chromosomal differences. Typically, outbreeding depression will happen if the populations have not had gene flow within the last 500 years. This is a rule of thumb only. It is kind of a shortcut. And this book says 500 years because it is from then that the human impacts have really fragmented the habitats in general. And if there is no gene flow within these years, it's because of human intervention, not because there was some adaptive thing happening before, okay? But if they did not have gene flow before 500 years ago, then maybe there was something going on there. So please draw from whatever source you can to see if there was gene flow happening. You can use historical records. You can use molecular approaches. Sometimes you will know managers have seen natural crossings occurring between the different populations. And yeah, it will also happen when you have substantial environmental differences. So the risk of outbreeding depression basically increases with the duration of isolation between them. But ultimately, even if one of these is true or kind of true, it must be weighed against the probability of your population going extinct. There are risks to doing nothing. So think of species that are very small. There's some in Europe, for example, in which there's only 20 individuals left. If you have literally only 20 individuals left, maybe you will consider using a different species. So there has to be some compromises. There can be some environmental differences. Ideally, you will choose a population that has the most similar environment. There can be some chromosomal differences, but maybe they are still able to breed between each other and have fertile offspring. And if some of these are unknown, keep going through the guidelines, through the decision tree, and proceed on an experimental basis. So you can try to do some crosses just for peace of mind in captivity or in glass houses, and make sure that, I don't know, the offspring don't have two heads or something. But if you did not find in your population that any of these three is true, you are looking into a very low risk possibility of a low risk genetic rescue. So you should proceed. We go back to the guidelines. Fourth step, we need to look at whether the benefits of augmenting gene flow will have sufficient benefits to justify the effort and cost of implementing them. And generally, the way you can do this is what will be the benefits of that. You will have the most benefits when your target population is very inbred. You will also see a lot of benefits when gene flow will be able to lower your inbreeding a lot. This will also be most severe in stressful environments. That's what we were talking about. Inbreeding depression is more severe in the wild. So if your population is out there in nature, you will likely see more benefits. And finally, the donor population is outbred. Preferably you should use one of those ones and more genetically different. That will bring in more genetic diversity and maintain evolutionary potential. And again, I sound like a broken record, but I really want to drill into you that the risk of doing nothing must be taken into consideration, okay? Maybe the potential benefits won't be as spectacular, but if the population is on the brink of extinction, it might be worth trying and keeping as much of the genetic variation that they have left. So yeah, we finished the tree and we have decided to do genetic rescue. Now, how do you go about actually implementing this? There's many things that managers must decide on. For example, how to augment the gene flow, when to start, from where to take the individuals or gametes, how many individuals, which individuals, how often, and for how long. So this section, I will give you some general, very general guidelines in two different cases, but in the case in which you have very limited information and cases in which you have a lot of genetic information. How do you go about increasing gene flow? This can be done in several ways. You can link fragmented habitats through corridors, for example, or you can move genetic material between them, so with translocations of individuals or gametes. Why are we doing this? Again, to recover fitness, so reducing inbreeding, and to increase adaptive evolutionary potential, increasing genetic diversity. So if we have this gradient in which at the top we have the less amount of information and at the bottom we have a lot of information, let's start looking into when we have little information. Maybe we know the effective population size, maybe we have an idea of how much migration there is between them, but there is not a lot of genetic data. First message, any gene flow into isolated inbred populations is better than no gene flow. Let's get that right off the bat. We have already gone through our tree, and we have already evaluated all the risks of outbreeding depression, okay? And we have determined that it's probably unlikely or that the risk of doing nothing is too high. So if your population is small and inbred, it's just a matter of time for things to get worse. And extinction risk will get higher and higher, and then everything will be more and more difficult. So the default should always be to augment gene flow. From where should you source immigrants? Well, populations preferably that used to be connected before and that got isolated within the last 500 years because of human impacts, that are not closely related. Preferably the donor should be outbred, they should have similar environments, and actually as many as possible. And I will come back to that. But how can you do that? There are some very cool techniques. Some indigenous, I think, American groups had some systems. So for example, in this system, if you have several populations, you go about in a clockwise way for gene flow. For the second generation, you do kind of this square system. And in the last generation, you go into crosses, like this star. And that will guarantee that the population will get as mixed as possible. and then you repeat again for the next generations. Okay, let's talk numbers. How many? How many individuals to move? And here goes the juicy advice. As a rule of thumb, one effective migrant per generation avoids fixation of alleles. In reality, in nature, because populations are not panmictic, because there is genetic structure and not everyone gets to reproduce, that kind of translates into 7.7 adult migrants. This is assuming that those 7.7 migrants will actually reproduce. 5 effective migrants per generation prevent harmful inbreeding levels. This translates into 38 adult migrants, 38 birds or 38 plants or 38 individuals. And as maybe you have caught it, but this is maintenance migration to avoid inbreeding. But more migrants will be needed if you already have high levels of inbreeding, if you want to reduce them back to a healthy level. And of course, the more inbred the population is, the higher the proportion of the population should be made out of migrants. So you can see that if you have 40% of inbreeding, basically half of your population needs to be from migrant origin. And finally, 5 effective migrants per generation are needed to maintain adaptive potential. So again, the 38 adult migrants. But it will likely be higher with climate change because environmental changes will be faster, will be more stochastic, will be more severe. So this is likely to be a low number. And again, if your population already had low adaptive potential, you will need that. You will need more than that. This is just maintenance gene flow. Okay, poll time. Can Bernd or Luis, can we have the third poll? For how long should you keep assisting gene flow into small isolated population? Ding, 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 ding. All right, we have 70 votes. Most people voted for the first one. Yes, kind of, and I will explain. First, I need to note that this gene flow needs to be done per generation unless connectivity has been restored. And when you say you did a corridor, you need to test that gene flow is actually going across. So yeah, you need to do it for as long as you manage to improve the genetic health. So these things that we were talking about, inbreeding and genetic diversity, have you improved that? Once you improve that, you will see that the population size will start to recover. It gets larger and larger. And that plus the connectivity that you already restored will eventually provide a self-sufficient population. But that's kind of the idea. We keep supplementing, we assist the gene flow until we leave them at a point in which they are self-sustaining, and then they can go on their own. Okay, second scenario, let's say we have genetic information. Again, in our gradient, maybe you have some markers, you have oral individuals, or maybe you have a pedigree, or you have basically everyone genotyped. As a reminder, we want to establish gene flow to maximize the retention of genetic diversity both within and across populations and to minimize inbreeding. What to manage? Well, one option is to minimize mean kinship. Mean kinship can be calculated from pedigrees or from genetic markers like SNPs. Yesterday and today with Laura, and yesterday with Kate, we saw other examples of how to go about it. And later in this session, Peter will tell you an example of how to do it with allelic richness. But mean kinship is the recommended procedure for managing threatened species. It has been used for a very long time to manage species in captivity, and it's also used in domesticated livestock breeds. So it has been really tried out for a lot of time. Okay, pairwise kinship, so kinship between two individuals, is a probability that two alleles, one taken at random from each individual, come from the same ancestor, so are identity by descent. So let's say pairwise kinship between individual A and individual 1 and individual 2. We have another level of kinship in which we have individual mean kinship, and that is the kinship of individual 1 against 2, individual 1 against 3, individual 1 against 4, and the kinship of individual 1 against itself. The next level is within population mean kinship, in which we average the individual mean kinships, all of them. And finally, we can have between population mean kinship. So that looks kind of like this. Let's say we have individual 1 and individual 2, and their kinship is 0.25. That is pairwise kinship. Then if we calculate all the kinships of individual 1 against the rest of the population, so against itself and individual 3, that is giving us the individual mean kinship, right, of individual 1. We can do that for all the 3 individuals, and we get their own individual kinships, and we average these 3 values, then we got the population mean kinship. We can do that for another population, let's say individual B. That would be within population mean kinship for B, and then we can do that across. So basically all the pairs across populations, that's between population kinship. Right. Why mean kinship? Why has it been so used, and why it's recommended? It is because the mean kinship of a population right now, it's on average the inbreeding coefficient that the next generation will have if they are mating at random. So if the present kinship is minimized, you will be minimizing the inbreeding of the next generation. Some people use kinship and relatedness as synonyms. That is not exactly right. Relatedness is equal to 2 times mean kinship, but that is only true if there is no inbreeding in your populations. If there is inbreeding in the populations, there has to be an adjustment, and there is a mathematical adjustment that you have to do. So mean kinship, low mean kinships reflect kind of the genetic value and the rarity of that individual. So the lower the mean kinship, the more valuable it is, because it is not highly related to the other individuals in the population. Closely related individuals, we have a higher pair mean kinship, right? So when you manage for mean kinship, you are kind of feeding 2 birds with 1 stone, because you will be both retaining genetic diversity, keeping all those rare alleles from the rare individuals, right? The ones with low mean kinship, and you will minimize inbreeding. Generally, gene flow should go from low mean kinship, so the valuable one, to the high mean kinship. The individuals are really related to each other. In this example, let's say we have 3 populations of about the same size. One of them has within population mean kinship of 0.01, 0.02, and 0.13. So this one would be the one that needs gene flow the most and the most urgently. From which one? From which of the populations? Well, you can decide that based on the between population mean kinships. In this case, we have 0.08 or 0.02. And again, as we said, the lower the kinship, the better. So we use this population. And if you happen to have a lot of genetic information for many of your individuals, you can go into more specific which individuals you bring. So you will bring the ones that are minimally related to the recipient population and that are themselves minimally inbred. This is just an overview. There are many ways to do this. There are a lot of softwares that help you to do this. Some use kinship, some use other variables. So we have PMX, we have Swinger, MetaPop2, and we are implementing some stuff in the dartR.captive. For how long? Again, should you do gene flow? Same as before, per generation until connectivity is restored. And the same principle of until you improve genetic health enough that the population recovers in size, there is connectivity, and then it's self-sustaining. Now, what if I do something wrong? What if I make a management decision and it was the wrong one? I want to ease your mind about this. First, we have seen that outbreeding depression is rare, and we know when it is likely to happen. Second, genetically swamping a population is hard. Genetic swamping means that you are bringing way too much gene flow, and you are kind of replacing the genetic composition of the focal population. It is really hard to swamp a population, actually. You require a lot of gene flow for that. We just saw that how many? 38 migrants, adult migrants, were needed to just keep retain genetic variation, I think, to not lose genetic variation and to not have harmful inbreeding. So it is pretty hard to do that. And generally, you know, if you're dealing with rhinos, if you're dealing with puquillas, if you are dealing with, it will be rare that you will manage to genetically swamp it. And again, and again, and again, you need to weigh the risk of doing nothing, okay? So in most cases, when you have highly endangered populations that are going extinct because they are very small, imperfect management is better than non-management. You must act. And as a peace of mind, you should keep monitoring. You should keep monitoring the genetics of how the generations are going about. And then we're working in an adaptive genetic management framework. So we're looking into these three, right? We are monitoring, we sample the next generation, we see how it's going on, we evaluate, and if we need, we adjust, okay? So last time for the last poll, please, Bernd and Luis. How confident do you feel now about starting genetic management? Let's wait for around 70 votes. Okay, good. I think it's generally better. We managed to have a positive impact, even a small impact or a larger impact. That's good to hear. And hopefully by the end of this workshop, you will feel even more confident because you will know a lot more. So thank you very much, guys. Maybe we can answer some questions.
Yeah, can we wait until the end for the questions?
Sure. Yeah.
Yeah, just want to make sure because we were a little bit late and see how we go time-wise. But please be sure you book Diana if you want at a table later or whenever she is available. You can use actually the people function and you can directly chat to people and invite them and make an arrangement. It's probably the best way because, yeah. That's the way. That's also Renee kind of was asked to organizing a table, and she best is to chat with her and get her directly when she's available. Okay. Thanks again, Diana. And I think I pass on to Peter. Can you unshare your screen, please? And then it's Peter Unmack now. talking about fish, I guess.
Doesn't need it. All right. I come. All right. Coming through. All right. I guess I can just do it straight. All right. Well, thanks, everybody. Thanks very much to Deanna. It was an excellent overview of things. I'm giving a very specific example, I suppose. I've been messing around in my head as to what's the appropriate way to mix populations and how many individuals based on the SNP data, and I started bugging Bern to write some scripts to do this. So this presentation is just a little bit of a summary of essentially things to consider, how to run the script, and then a little bit on how I interpret the outcome of those analyses. So essentially, in my case, normally I've got a situation where we've got a threatened fish species, we want to bring it into a hatchery or some sort of a breeding program, and the question comes about what populations should be used and what proportions from different populations should be used to establish a hatchery stock. Now, in some cases, it may be inappropriate to mix populations, but in most cases, it probably is, as Deanna has outlined really well. And the broader goal usually being we want to conserve genetic diversity, but you're always constrained by the reality of managing the hatchery and limited resources, both limited resources in terms of how many fish you can hold, but also how much data you can collect and how much monitoring you can do post-breeding and stocking. There we go. Now, there's multiple philosophies that exist on what sort of things to consider when mixing populations. So the one from the Frankham and Sunnick's books is, focus on using less inbred populations, but inbreeding can be a little bit tricky to measure and compare between populations, especially if all of your populations are already a little bit inbred. It can be a little bit challenging. They make the key point not to focus on rarer genetic variation, as it's most likely deleterious. Most new mutations are likely to be, worse for you than better. So they argue that including rarer variation can be a bit more risky. And of course, Deanna had both their books there, but excellent reading for anybody considering working in this area. Some other folks have kind of argued that just targeting adaptive loci is the key thing to go for. We recently had Fred Allendorf visit ANU and gave some seminars, and some of this sort of stuff was discussed. And so there's definitely a school of thought out there that focuses on that aspect, but it's not always easy to identify adaptive loci, especially when you don't have the resources to go figure out what they may be. And it can be quite challenging. One of the hard things with the SNPs, of course, is you're mostly probably measuring neutral variation, which isn't going to necessarily affect the outcome of what it is that you're mixing. And it's generally a little bit tricky to identify ones that are under selection. So what do you do? So essentially my take on this sort of stuff is that all we can really do is treat SNPs as a proxy for important genetic variation. So the idea is if you can conserve the SNP variation we observe, then you're likely conserving any other important variation that we haven't measured. Obviously, this is far less than perfect, but it's not clear yet that a better solution exists for this type of data. Now, if you're doing whole genomes and you can do all these different experiments and all this other stuff, well, then this doesn't necessarily apply because you are collecting much better information on which to base your decisions, but we rarely have that. So this leads to a whole bunch of challenging questions, and I think, I liked how the end of Deanna's poll had now I'm just confused and overwhelmed. If you're not confused and overwhelmed, you're probably not doing it right because you're basically playing God, but you don't know what the outcome is. And then you have to justify why you're doing certain things to lead to that outcome. And it is really challenging because most times we don't know, but this is our best guess. And so that some people are better able to deal with that than others. But that's the real challenge with this sort of stuff is you've got all these decisions you need to make and it's not always straightforward as to what the right answers are. So, you know, how rare does an allele have to be to say ignore that allele? And of course, there's no definitive answer to that. But I think just about everybody agrees that most rare alleles are probably irrelevant and can be ignored. But, and Deanna kind of covered some of these things a little bit, you know, how many populations to include in your mix? Some of that can be political. So if it's a New South Wales hatchery, it's often challenging to get fish from Queensland, different legislation, all kinds of other issues. So often they stick to stuff within political boundaries. How genetically divergent do populations get before you say we won't mix those populations? And that's another really challenging question. One of the things that can kind of potentially give a bit of insight into some of that stuff is natural introgression between populations. So you can actually get introgression between quite highly divergent species that actually may potentially lead to good outcomes. So there's nothing that says just because they're divergent means you don't mix them, but it's just another part of what you have to consider as part of that. You know, and how much you worry about potential local adaptation, it's probably been a little bit, we've probably been a little too overly concerned with that in the past, I suspect. But there's so many different ways that something can become locally adapted too, and it's really hard to measure the broad spectrum across a species biology as to what's an adapted trait and what's not. So you can match critters to similar environments. If you're stocking them into a certain place, take them from a similar place. So that's one way of potentially getting around that a little bit. But, you know, it depends whether you focus on temperature or habitat or, you know, there's so many different things you can focus in on that make that pretty challenging to worry about too much. The other alternative is just say throw them all in a bucket and let nature sort it out. So the main risk with doing that appears to be, you know, breaking up co-adaptive gene complexes, which obviously, if they exist, can be extremely important. How common are they, and how easy are they to identify a priori? And so, you know, I don't really know the answer to those questions, but it's not always simple to know these things beforehand. How long ago a population like diverged? You also need to consider sample sizes. One of the things when you're doing this sort of stuff, it's really nice to have balanced sample sizes across the things that you might be comparing. Often in reality, you don't get balanced sample sizes. Sometimes we have 1 or 2 from a population or 20 or 30 from another population. And that's just the constraint of working with what samples you have on hand. But it's important to think about, as you do these analyses, about comparing unbalanced to balanced sample sizes, because it will massively affect allelic richness, which is the main thing this is based upon. And yes, there's a whole pile of practical constraints as well. It's fine to come up with a number that says we need this many individuals per population all mixed together, but there's no guarantee they're all going to breed in that ratio and all going to contribute equally to the next generation in that same ratio. And there's also going to be massive constraints on how many individuals a facility can hold to breed, or perhaps how many you can take from the wild if the species is really rare. The other thing to consider is SNPs are only measuring a part of the genome. I hear the figure bandied around 1%, whatever it is. So you've also got to keep in mind that you might say, okay, 20 fish is enough to capture almost all of the genetic variability, but that's only for that 1% of the genome you've actually measured. So you've always got to keep those numbers as not as your bare minimum, but it's more about the ratio. And obviously you want to bump those numbers up as high as you can based on what you can hold and breed. So again, Deanna kind of made this point. I'll make it maybe slightly differently. And sorry about the death by PowerPoint here with too many words on the slides, but I didn't know how else to do this. But we're in a biodiversity crisis here, and we don't have the resourcing to really pull ourselves out of this. There's too many species in a bad predicament, limited funds, limited people to research them, and there's no real straightforward way that you can do it perfectly. So you can aim for perfection and conserve 10% of the species maybe, and then leave the other 90% up to just chance as to whatever goes on in the wild. Or you can try and conserve 50% up, make some mistakes, screw up 10% of them, and then the rest are left to chance. You know, I think we've got to change our thinking around conservation biology to say We will make mistakes, and it's okay to make mistakes. You hope to learn from those mistakes. But if we don't do anything, these species are probably screwed, at least some of them. And if you do something, we might mess up some, but they're probably screwed anyway. So at least you might be helping some, you might mess up a couple, but overall, hopefully, you're providing far more benefits than just doing nothing and leaving it all to chance, because chance will eventually eliminate them. The other thing, in the case of a lot of these fish, we can often conserve the founding populations, mix them in the hatchery, and then stock them into new places. We're usually not putting fish back on top of existing populations. So if you make a mix and it turns out that was a bad idea for some reason, you can go back to your wild stocks and start again, or change the approach you're doing. Now, you don't always have that possibility, but if you can, it's a good idea to keep that in mind. Now, there's a lot of different ways you can process your data in how you analyze it. For some analyses, we hate missing data. We want great loci, every little detail as good as we can get. For this sort of stuff, it may be a teeny bit less important. You know, filtering less hard, you might actually obtain more loci, although I don't worry too much about the exact number of loci, but the point is you can probably relax certain parts of the filtering. For reproducibility, I tend to go with what the Dart folks suggest, which is 0.96. A lot of people filter at 0.99 or 1, and that's going to eliminate some loci that may actually be quite good, but don't have quite as high a reproducibility for various reasons as part of the pipelines and how well things sequence. Read depth is another kind of tricky one, and I don't really know how to interpret what read depth to say is too high. Certainly at low read depth, you get problems due to samples that have not sequenced as good as other individuals. And so that tends to bias you towards missing real variants. So if you want slightly more stringent data, you know, you might want to do your low read depth at 10. And then depending on what sort of curve you see in your data when you run the report for read depth, you know, how long's the right-hand tail? A lot of the times you can clip off the majority of it without losing very many loci. The idea being that the ones with very high read depth are more likely to be duplicated loci that have slipped through the pipelines. We've never verified any of this, but that's just how we tend to think about things as we go. So more often than not, missing data by individual is typically due to poorer quality DNA that just didn't sequence as well. And that results in an underestimate of heterozygosity, and you get fewer variable loci. Whether you remove these individuals or keep them in, it may not affect things all that great, because if their genetic diversity is a bit low, leaving them out may not have that much influence. I usually tend to remove them just to try and keep the individuals in the data set a little bit more consistent if possible. Again, I kind of said missing data by locus. You know, it depends on the data set. Sometimes, you know, some data sets, you know, 0.5 has an awful lot of loci, and any filtering above that removes a bunch of loci. So it's a balance between, you know, how much you're able to compromise, that sort of stuff. And of course, you run the analysis with a lot of missing data or very little missing data, and then compare across those outcomes. Rare alleles, for a lot of the more threatened fish that I've worked with, rare alleles often quite dominate the data sets. In some cases, up to 2/3 of the alleles will be removed when I take out the rarest 2 alleles. And if you bump that up to a higher value, you don't end up with a lot left. But it really depends upon, you know, your sample sizes per population, your overall N, how fragmented populations are, distribution of variation within versus among. But by default, I normally remove the rarest 2 alleles. So alleles found in only 1 or 2 individuals in the data set. Now, most of my data sets usually have between 100 and 400 individuals, to give you some idea. And so if you've got more individuals, you want to bump that number up to higher. The last really key point is, if you've got any individuals there that are artificially too heterozygous for either contamination or whatever reason, we see a few in most data sets, you've got to take these ones out, because they will always be picked first by this method. And they're artificially inflated and you don't want them in there. I usually export or run a quick heterozygosity command to look at how that varies across individuals, across the entire data set, and also by population. Most times you can identify them from PCOA plots. You'll get a nice shotgun blast of your population. There'll be one or two animals away from that central point. And if you look at them, they'll usually have high heterozygosity or very low heterozygosity. And often when you put those individuals into a tree, they'll be the first branching individuals of that lineage. So there's other ways you can identify them as well. What I tend to do is when I first run the script, I usually just check the first few individuals it's picking out just to make sure that they are normal looking individuals within those populations and don't have obviously excessive heterozygosity. And it's hard to know, what makes an individual too heterozygous? Well, that's a bit of a continuum. Sometimes you'll see a very clear jump where 9 of your 10 animals from a population might vary by 0.1 to 0.2, and then you've got one that's 0.4. And you say, well, that 0.4 is double the rest. It's probably wrong. And I take those out. But it's not always super clear cut. I think one of the things we may probably need to think about with this script and this sort of analysis is trying to pick more average individuals to choose from rather than individuals that have unusually low or unusually high values across the population. Normally when I do this, I end up doing a lot of different mixing analyses, exploring the effects of different sample sizes, unequal versus equal, different filterings, different numbers of rare alleles. And the idea is to see how much does the proportion of individuals suggested for the mix change between these analyses. And they almost inevitably do, but it's kind of all about data exploration, figuring it out, and then understanding what's going on between those comparisons when they do differ. So the little bit of data I'm presenting today is for purple-spotted gudgeon. They have been in long-term decline in the Murray-Darling Basin. They're quite easily bred in captivity. They do really well in smaller habitats. They're commonly called mogs. The scientific names are Magurna adspersa. They've just been listed federally under the EPBC Act as a endangered population of that species. So essentially within the Murray-Darling Basin, see if we can see my cursor here. So these little circles just represent regions where there's still extant populations. So this is like one population. This is one. There's 3 or 4 up here, I think. Some of the ones with an X on top are ones that have gone extinct. Most of the extinct ones are gone. We have representatives from Murray Bridge still in captivity. But that's actually, so this fish would have once been widespread across almost all of that portion of the Murray-Darling Basin. So extremely fragmented into tiny little populations. And a couple PCOA plots just to give you an idea of what that looks like. Most things separate the southern Murray-Darling thing from the Loddon and the lower Murray from things in the Darling River here. And then this is just the Darling catchment. So this is what I'm going to be focusing on for the little bit of data analysis. So these things separate nicely. This Army thing is a population that was admixed in the late 1990s. And it was a mix between fish from the Guida River and fish from the Condamine River. Now, I've got a McIntyre and a McIntyre 2. So everything from Tenterfield Creek groups in with Condamine fish. Everything elsewhere in the McIntyre system is a little bit different. So that's just why I've distinguished between the two, because they're within the same river basin. And this is just some basic stats, just showing some diversity measures and whatnot. So you can see sample sizes vary from 9 to 79. The number of polymorphic loci, P-lock, varies quite a bit, and that's often tightly correlated with the sample size, which is why you've got to think about that part of it. Proportion is the number of the total loci found within that population. And then HO, HE, some of these do vary a bit. So for Condamine fish, there's 3 populations in there, and that's why your HE is a bit higher, just there's a bit of differentiation between some of those populations, which throws out the HE because they're not a freely intermixing population. Fixed alleles, fixed differences are fairly rare. Macquarie has some, and there's a few private alleles. And note that this data set that I've just presented is a little bit different to the example I'm using, but just to give you a feel for what that looks like. All right, so I think at this point we're probably ready. Bernd was going to work through. So the initial data set's already been filtered for reproducibility at 0.96, read depth of 100. Any funky individuals have been removed. All right, and so from there, I shall unshare my screen. Pass to Bernd for a minute here, and I'll probably both talk a little bit maybe.
Yeah, and I probably talk over there. Hopefully you can hear me because use one microphone together. And I guess because of the time, it's you can't hear me? Okay.
Can you? Yeah. Okay. Cool. No. Okay. We have Chin doing that. Well done. Thanks. So okay. So I talk now, and I think just to make life easier, I will not expect you to follow that because we are a little bit behind schedule. So we just demonstrated. Is that okay?
Yeah.
All right. So let's go to a beautiful screen. Yeah. That's basically they can kick back and relax, and it's all good.
Your camera.
Yeah. Okay. And here is my beautiful camera and my face. Okay. So we go to small population 6 and make sure if you really want to follow, there's a session 5.6 now in the PopGen master. So you need that. Otherwise, you have the non-updated version. But as I say, just kick back and relax and enjoy the show, I guess. So this is the data. It will be also on the website soon. And basically what you see here is the script. And we do the usual thing. We load the packages and the data. And then Peter is subsetting it to a certain numbers of individuals from the darling.
Yep.
You want to talk or I talk?
No, you talk.
Okay, so I try to understand what it's doing. Then obviously he filtered for, as he said, quite a few filter steps are there. Filtered for the monomorphs, loci, obviously because they don't do anything except make things slower. And then he filters for loci, quality in that sense, for the missingness, call rate for 0.95, so pretty high. So get rid of all loci which are below 0.95. And then he has this beautiful function, which probably most of you haven't seen.
So maybe just pop back up. So the key thing there is there's not that many loci under 0.95. So hence I filtered a little bit more stringently in this case. Did that come through?
Okay, good. And then he uses this site frequency spectrum command, which gives you basically just a distribution of allele frequencies. And we have some expectation. We come to that tomorrow in session 7, but it's a nice precursor. Well, that's the allele frequency distribution. And if I may explain, or you basically, you look at all your individuals and you see how often an allele is found. And you have lots of alleles which are found only once or twice or 3 times. And obviously you have more of them. And you have not many alleles we are found and basically in all of the individuals or in 50% or something. So that's the distribution of the allele frequency called, for whatever reason, site frequency spectrum.
So that gives the numbers.
Yeah, and obviously this function is probably not really nice, but it gives you here the numbers. Basically, we filtered for monomorphs, and that's why we don't have a D0. So we don't have any allele that is 100% in the data set anymore. Yeah. Okay. Then we filter for minor allele frequencies, and again, this function is a bit funny. If it use a number below below 1, you filter for a minor allele frequency. If you use basically a number above 1, if you use a whole number, then it filters for how often this occurs. So if you would run this SFF command again, everything from 3 would have been gone.
So
only
D1, D2.
So D1 and D2 will be gone. So that's for minor allele frequency, in this case, counts, allele counts. And then You want to explain it or shall I? Fun?
You keep going.
Okay. And then we have a new function which is called GL opt-in all individuals. It's kind of the idea is the following. How many individuals you need, which is on the x-axis, to retain a certain amount of alleles. So you take one individual, and the first individual gives you something like 60% of all the alleles. Then you take the second allele, second individual, and then you have normally 70%, and then 80, and so on. Obviously, you take all of them in the population, you get to 100%. So this basically tells us, and you do that basically in a randomized bootstrap approach, and it tells you from which population you get an individual to increase the numbers of alleles, and that can support the decision how many individuals I need from which population to conserve the diversity.
Graphical representation.
A graphical representation of that, yeah. And obviously you can do it a little bit nicer in terms of analysis.
So maybe just explain the subsample script, because that's the only difference in the second run.
Okay. The idea is then obviously we have this.
So the first run was all individuals.
Right.
The second run balances sample size.
Okay. You want to go?
What do you say?
Okay. So basically, obviously, the sample size in each population is different. We had that, and we want to make sure sample size is not affecting our populations, kind of which individuals from which population you want to use, and therefore we do kind of a bootstrap approach and subsample our population to a certain number, yeah. And this is a special function. It's new because some of the population actually don't have this amount of individuals. So we had to hack the function. So make sure we take 14 if we can, and if we don't, we take what we have. And again, we filter for all of the same thing as before, for the subsamples now. And then, you want to say that?
So I can show some of the stuff in Excel if we like, just to.
Yeah, okay.
Good. All right. I'll just quickly show people how this looks in Excel. Just time screen. All right. So basically, I just open the CSV file in Excel. You get these first 5 columns. Now, Bern can fiddle around and do it in R. I'm not that good in R, so I use Excel. And essentially, all I do is I just go. I select the population. So the last column is the percentage of alleles you've captured. So basically, I take capturing 90%. I highlight those, and then I just insert a pivot table. You can pop it on a new sheet or an existing sheet, it doesn't matter. And then you just click that, and then mine's set so that it's to count pop. So that basically just gives me the number per river that it's selecting from that mix. And then I do the same thing down to 0.95. Again, it's a bit arbitrary whether you want to go 0.9, 0.95. These are there's no set rule. I'll just do the same thing again, pop it there. Right, so then I generate that, and then what I do is I just copy that, paste special over the top for values, because then I can easily manipulate stuff. And essentially that's the table up above, and then I've just added this function in, which compares the number from that river over the total number to give me the proportion. So you just use the $ in Excel to indicate that that cell does not change when you drag it to change those values. So what does this look like? So what I've essentially done here is, so the first one is the one with all individuals. The second one is with an average of 14. So comparing, so what we really want to understand is how does the proportion change? I'm not too worried about the number of alleles it's conserving, whatever, it's the proportion that matters, because that's what goes into the hatchery. So here I've just done a simple thing where I've just subtracted the count from 0.95 from the count from 0.9. And basically, because there's about 10 individuals, 0.1 is one individual. So we can see, for the most part, you know, condamine changes a little bit from a proportion of 11 to 0.25. One drops 0.13, but the other three don't change very much. Same thing for the second one. These really don't change as much at all. Slight difference there at 0.08. And then I've just taken this column here. Let's see, which one did I subtract first? Yeah, C3 from C2. So I've just taken the uneven sample size and then subtracted that from the equal sample size. So you can see most of these vary by 1 or 2 individuals across the populations, and then same at 0.95. And these are actually pretty similar. So, you know, one suggests maybe one extra individual, one less individual, but they're not too different to each other. But that's kind of how I take some of these values and look at them and think about them in terms of what some of the outcomes might be. Now, let's see, let's just pop back here. We can pop through those. Yep, so then I take the Excel, that bit. So that's the general gist of just how to run the script, how I look at the data within Excel. You can do all that table stuff in R if you're clever enough. So part of the reason why I like to do this on many, many different variations on the data is to get some handle of what's affecting the numbers you see that the different analyses suggest mixing. So if sample size doesn't matter, then you won't see any difference between a standardized sample size and an uneven sample size, but you would expect to see some differences. If you change the filtering, again, you can compare stringent filtering against less stringent filtering. There's many different aspects of that you can tweak. So it's really up to what you think is really important as to what you're comparing and contrasting. For me, the key outcome is what's the key proportions, right? We're not saying, oh, you only need 6 fish and you conserve 90% of the genetic diversity. That's not really true. But hopefully you've got a hatchery situation where you can bring in 50 fish and you can bring them in that proportion into the hatchery to try and get that balance across those populations. But you've got to keep in mind that animals in hatchery or anywhere aren't going to breed in the proportions you've designated them to breed in. We just hope they will. And of course, you can also get unequal amounts of contribution from the various pairs and things that you mix. So it's not a perfect system. It's really just this is kind of the best guess to get hatchery started in the right direction. It's not necessarily, it's a long way from perfection, but it's a lot better than probably just taking a random guess and saying just do everything equal. At least we've got something to stand on to justify why we're not just taking an equal proportion. The hard part is for most of these things, you know, governments just don't have money to spend to do the proper stuff you need to work out what's the outcome of these mixing events. How do you improve them? How do you make them better? The thing I would emphasize to anybody doing this stuff, the most important thing, you need the samples from the animals that went into the breeding population. You need the samples from the animals that are breeding in the wild. It's very cheap, very simple usually to collect some genetic samples. Build up enough samples, write an ARC grant, get the resources to then properly evaluate this stuff. But if you don't take those genetic samples at the start, you're never going to take that further than just guessing. Now, how much of this stuff is going on? So there's a fair bit of work in multiple states. So South Australia's been going for probably 2 decades with some of their breeding programs. New South Wales is doing a bit. Victoria's been doing some. This is part of a program from the North Catchment Management Authority in Victoria with Peter Rose, where they're bringing back the Magnificent Six. So they're actively collecting wild fish, breeding them in hatcheries or in dams, and then stocking them into other places, and doing that with an eye to genetic mixing. In fact, we were just out last week with one of Elisa's PhD students collecting pygmy perch to assess some of this stuff. So we're kind of at the forefront of doing this. I don't think it's been going on for that long, but I think we're kind of at an interesting point where we'll start getting better answers as to how well we're doing at playing God and driving better outcomes and getting better genetic diversity into these populations, rather than just allowing all these individual populations with different alleles just to blip out and be gone. And then I'm just showing off at the end with a big Murray cod I caught Cataract Dam last year. Anyway, I welcome any feedback. It's in some respects, it's not rocket science, but as we've kind of stressed, there's a lot of unknowns, and it's challenging at times to wrap your head around those unknowns. Sometimes you just got to jump off the bridge and hope it's deep enough. Thank you.
Okay, great. Thanks, Peter. Thanks, Jana. That was excellent. As I said, there is the tutorial is running, and there is some caveat, but it should be okay. I want to show one bit because from now on, hopefully we sorted this POSIT cloud thing. So the last time I say something here. So from now on, when you log in, you will see, and I share my screen this time without talking without screen, and you should see always something. Yeah. So I logged in now as a normal user, and that's what you see next to yours. There will be always a master Jedi, and it has a session number to it because I have to update all the time because these presenters, they're just sloppy and come late in. So basically, every time we have a just ready. So tomorrow there will be a master session 7 to 9, I hope. So please make sure when you come in, go to dartRverse and then find the latest version, and then this works. For example, the opt-in function, it's a new one and it's actually hidden, so you can't see it, but it's not like we want to hide it. It is there, and you could even copy it from there. It will be hopefully in dartRverse soon. Yeah, so for the latest one, go always there, and it's fine. And I think I stopped there because you have seen enough of these kind of things. Basically, you log in, and as you can see, there's a temporary copy. Just make your copy, and then you have the latest version and can do whatever you like with it. Okay. Questions? Do we go for it? Yep. Are there any questions we know for Diana and Peter?
Are
there? Okay.
Yeah, at the top of the chat, there were several questions.
Okay, I put them on. Sorry. This one?
What's that, Chris?
Thank you. Right, question from Chantel. Metrics suggest. Okay, yeah, generation times are long, populations are limited. Yes, well, generally the metrics are the ones that we mentioned, right? To identify genetic erosion, the same inbreeding, allelic richness, kinship, the usual ones. And well, the fact that it has long generation times means that gene flow will take a long time to kind of come in, and it will be slow for you to see the benefits of it. Yeah, so I do recommend starting as soon as possible. I guess they will have probably overlapping generations, so yeah, you can start from the beginning, and you can start early on, and then you will see it eventually until it reaches most of the population. Yep, next one, please. How do you distinguish a small endangered population with inbreeding from a new population due to range shift originating from few colonizers? Right. Well, I guess that they will have a lot of similarities. So I suppose that this is kind of contrasting two scenarios in which there used to be a range, a large range, and then suddenly all kind of shrunk, and you had some isolated populations. That's the first one, versus a new population that kind of started from one, and then you have like a subsampling, a smaller one that is created from some colonizers from that big one. They are likely going to behave genetically very similar, so we'll have more or less the same problems: inbreeding, loss of genetic diversity over time. And I guess the thing that you need to keep the most in mind about managing them is the one that is a derivative. You should be careful to not bring gene flow from the mother population, because they are just a subset, right? So most of the of the genetic variation that you find here will already be in the small one. If you have other options, try and choose the other populations to bring some new, fresh genetic variation. There's no silly questions. Right. Good point, Nirjana. I guess that genetic erosion is like an umbrella term to say that the genetic health of the population is not going well. And by genetic erosion, we mean different things. One of them, different metrics, right? Different components of genetic health. One of them is genetic diversity. The other one is inbreeding. The other one is effective population size. So I guess genetic erosion is kind of like this big umbrella term that encompasses many things. We are waiting for a question, I think. Okay, question from Lerato. How about the risk of introducing maladaptive alleles from potential donor populations, especially if you didn't catch it during analysis, to the at-risk population? That is a great question, Lerato. I think there's a mic on. That's a great question, Lerato, and I'm really glad you brought it up. I guess this is one of the topics that has been really hot lately, and that is genetic load. There's huge conversations going about that, but I guess the gist of the problem is that generally it took a long time for scientists to kind of convince managers that bringing genetic variation was positive. And we were doing some process into that. Then there was a stream of publications that started saying that that can be dangerous because you might be bringing in genetic variation that is not useful or that is harmful into the small focal population that you are trying to save. And to those publications, there has been responses for those ones. And those responses have systematically addressed all their points and systematically challenged them. So if I share my screen, can I share my screen? Okay, I cannot share my screen. We are busy. Okay, we can share my screen. Perfect. Yes. Can I finish this answer? Can I finish this answer?
Yeah.
The one I am at right now.
Okay.
Okay. Yeah, I will finish after this one. So these three publications have already challenged some of those cases. Yeah, please take a screenshot, please read them. But basically, the message is that the importance of these harmful alleles being introduced has been overblown. They are likely not going to have any effect. And that is because they are rare, you are bringing them in, but you are also managing for inbreeding. So most of these rare alleles are recessive. And if you are managing for inbreeding as well, they will not be expressed. However, rare alleles, and this is something in which I disagree with Peter, I do think rare alleles are important to keep in the population because they increase evolutionary potential. So right now, they might be harmful, or maybe they have no consequence, but when conditions change, they might. And the more diverse your toolbox is, usually the better. The more chance you will have, the population will have to adapt. Yes, so that is my answer to that.
can see something.
Yeah. Got to get this share screen up first.
No.
No.
You're almost there, but just I kick in. Diana will be certainly available on a table. There's so many interests. Diana, when you want to do your table? Okay, after say, when I was at say 4 o'clock, you're at the table? So I say again, Diana will be at a table at 4 o'clock, somewhere in the Jetty chamber, and Bill is now answering a question.
Okay, got it. So that's a bit small. You might need to enlarge your screen. There's some arrows there. Sorry, I wasn't prepared for this. So the question about what's the difference between genetic erosion and inbreeding? Well, here's 3 different populations which show that those 2 things can vary completely independently. So population number 1 has got 3 alleles, A1, A2, and A3. And it's a completely randomly mating population. So it's got 3 alleles, it's got high heterozygosity, it's got no inbreeding. Population B has got also no inbreeding, but it's got lower genetic diversity. It hasn't got allele A3. So only got A1 and A2, and also they're heterozygotes. Population number C has got all 3 alleles, so it's got quite high genetic diversity, but it's terribly inbred. If you look along the top line, there's just a whole lot of A1 individuals breeding with each other in a family group, and so they're not producing any heterozygotes. There's another family group of just A2s, and another family group of just A3s. So that's severely inbred, but it has the same genetic diversity as population A. So the inbreeding can vary quite independently of the genetic diversity. And this one has different genetic diversity, but both A and B have no inbreeding. So the 2 things can vary completely and utterly independently. And as Diana pointed out, when you get both of them at once, genetic erosion and inbreeding, then that gives you really low heterozygosity, and that's a bad thing. But the separate management strategies for how you stop inbreeding, as I mentioned earlier this morning, you might introduce a pollinator that went extinct if it's a plant that's getting inbred, or you might, if it's a animal species that's having trouble dispersing because of an introduced predator, you might manage that predator. So that's how you manage the inbreeding by non-genetic methods. Either of them can be managed by managing the gene flow. The genetic erosion can also be managed by other methods like equalizing family contributions and so forth that I talked about before. But I hope it's now clear for the person who said, I can't see how inbreeding and genetic erosion are different things. Those 3 populations have completely independent variation of those things. Thanks.
Thank you, Bill. Okay.